In agriculture here in the UK and actually throughout the Western world, this was a very familiar sight. Very intensive agriculture, heavy use of automation, of tractors, etc. And in this case, a heavy dependence on chemicals, among them chemical fertilizers that drive food production. And if we look at global nitrogen consumption around the planet, you can see that the developed world are very heavy users. This is just a map showing the global nitrogen consumption around the planet, and you can see that the developed world are very heavy users of nitrogen consumption in their agricultural systems. Now, in contrast, when you look at Africa, you can see that it's really limited in its nitrogen consumption. Very small amounts of nitrogen. Actually, most of the countries where you do have some nitrogen use, such as Malawi here, is because the government chooses to subsidize nitrogen. So in most cases in Africa, the farming system is a smallholder farming system. These are people with often less than a hectare of land, very few financial resources, and really do not have the capability to purchase the fertilizers to invest in their crops. So in many ways, the nitrogen issue is we have two dichotomy. We have a dichotomy in the nitrogen issue where you've got heavy use in the developed world, with all the environmental consequences of that. And the use of nutrients in agriculture is a major cause of pollution coming from agriculture in the developed world. And in contrast, most farmers in the developing world have very little access to fertilizers, and as a result, the yields of their crops are very poor. And indeed, in this analysis, which was just looking at what limits food production around the planet, if it's in green, it just means that it's at least above 50% of the yield potential of that region, so it's not in the analysis. And here they're looking at two factors, water and nutrients. Now, and this is just for maize production. So in this paper, you can go and they do all the cereal crops, maize, or at least a majority, maize, rice, and wheat. And if you look at all of the major cereal crops, it's pretty much the same across the globe. So you would have thought, particularly in Africa, we often think of Africa, and often when we see Africa in the news, it's because they have a drought. And so the food crops have failed, people are starving. And we often think, therefore, that water is probably the biggest limitation to food production in Africa. That's actually not the case. So Ethiopia is having a drought right now, but they haven't had a drought since the 1980s, or nothing particularly severe. And in the 30 years in between, they've actually had sufficient rain to produce their crops. So yes, they do have droughts, and a lot of countries have droughts. It's just that they don't, if they really don't have the resources, or they haven't had the resources in the 30 years when it was raining enough to grow enough crops that when a drought hits, they've got the excess or the financial security in order to see themselves through that drought. So in fact, when you do the analysis on maize production in Africa, it's not water that's limiting maize production, it's nutrients, and that's nitrogen and phosphorus. So in red, it means it's just nutrient limited. There's ample water available from rainfall. And if it's blue, then it's water and nutrients that are limiting. So it's only really some parts of the more arid areas of Eastern Africa where if you want maximal productivity, if you want to gain at least 50% of your potential yield, you need to irrigate and you need to fertilize. Around the most of the world, it's fertilization that's a limitation to food production in the areas that are not achieving their, or even close to their potential food production. So in recognition of this, the Bill and Melinda Gates Foundation agreed to fund a program called Engineering the Nitrogen Symbiosis for Africa. Now, obviously, if I can engineer the nitrogen symbiosis, it has potential across the planet. But for me, I'm focusing specifically on the smallholder farmers in Africa who really suffer because of the inavailability of nitrogen. So there are multiple ways to solve this problem. I fully acknowledge there are different approaches you can take to tackle the nitrogen problem. And I'm only taking one approach. And I do not exclude the possibility that there are alternative approaches that one can take. And I think the nitrogen problem is such a severe problem, it's so intrinsic to our whole global food security, that I think it's entirely justified to say that you should set up multiple programs running in parallel attempting to solve the nitrogen problem. And it's possible that associative bacteria may solve the problem before I can do it. that's fine. If somebody can solve the nitrogen problem, I'm very, very happy. So my research is focused specifically on the symbiotic associations that legumes form with nitrogen-fixing bacteria. For the reason I choose to focus on this is because I have a working prototype. Here we have a system that works perfectly well for solving nitrogen production for the plant. The legumes form these nitrogen-fixing nodules on the roots, and the bacteria that reside inside those nodules can supply all of the plant's needs for nitrogen. So I like it because it's a prototype. I have something that I know is working, and if I can transfer that capability to cereals, I know I have something that will work. But as I said, I fully acknowledge there's other processes to do to solving this problem. Now, I just wanted to just show this graph to sort of map out the opportunities in this space. So world average nitrogen application rates are around 130, 140 kilograms of nitrogen per hectare. In the UK, we're somewhere around here. In China, they're actually off the graph. A legume fixes nitrogen generally in excess of 100 kilograms of nitrogen. On average, it's around 120 kilograms of fixed nitrogen per hectare, but you can have legumes that fix at higher levels and legumes that fix at lower levels. But there's a range in there. But on average, around 120 kilograms. So that rate is really at the top of this graph. So if I want to replace fertilizers in totality on the planet, I have to get the full complexity of the legume symbiosis functioning in a cereal. But actually, for smallholder farmers are applying extraordinarily low levels of nitrogen, somewhere in the range of 3 to 5, in some countries up to 10 kilograms of nitrogen per hectare. And we're looking at few percentage of the levels of nitrogen, and that's the totality of the nitrogen that they apply to their fields. And a lot of that is actually coming from their own manure that they produce in their household. A lot of smallholder farmers don't even have the domestic animals to produce manure to fertilize the fields. So here I think there's a real opportunity because actually relatively modest levels of fixed nitrogen have the potential of doubling yields for smallholder farmers. So of course I aspire to get over here to replace fertilizers in totality and to get to the sort of maximal yields. But I acknowledge that's a very significant challenge. And if I can get even modest levels of fixed nitrogen in context to what a legume can fix nitrogen at, then it has the potential to be quite transformative to those smallholder farmers. Now, the challenge that I face is the complexity of this system. It's an incredibly involved process, the nitrogen-fixing symbiosis in legumes. So the bacteria recognize by the host plant and form these root hair curls that entrap the bacteria in the root hairs. The bacteria replicate inside those root hairs, and you get these unique structures called infection threads that allow the colonization of the plant by the bacteria. And we really don't see these structures in any other process except in nitrogen-fixing symbioses. Associated with this colonization of the host plant by the bacteria is the initiation of nodules, which start as cell divisions in the inner cortex, and the development of the nodule, the cell divisions that occur in both spatial and temporal coordination with the bacterial infection processes, such that these infection threads, which you can see, grow down and ramify through the developing nodule, and ultimately the bacteria are released from these infection threads. So this is a cross-section across a tip of infection thread, but one that's down in a cell inside this nodule. And you can see the bacteria being budded off from this infection thread and released into the cells of the nodule. And ultimately, if you take a cross-section through a nodule, this is a soybean nodule, you can see this is an uninfected cell, and these are two infected cells either side. And you can see these infected cells are completely packed full of these bacteria, always contained within what we call symbiosomes, and so that's always surrounded by a plant-derived membrane. And in many ways, that you can think of them as nitrogen-fixing organelles. Nitrogen-fixing organelles that evolve through this symbiotic symbiosis, this symbiotic association. So these are difficult processes to engineer. And how do you transfer something as complex as this from a legume into a cereal? And that is my challenge. So I've feel like if we're going to start tackling this, you have to break it down into multiple achievable or more achievable processes. So I think there's really 4 major steps that you require to transfer to the cereal to get efficient nitrogen fixation. First is to get the cereal crop to recognize the rhizobial bacteria as a potential beneficial symbiotic organism. You then also have to engineer nodule formation. Although I don't think the nodule in many ways, I think, is an amplification of this process. So I think what you really need is a reasonable amount of cell division happening in the cells that are going to accommodate the bacteria so that you can amplify the number of cells that are fixing nitrogen. This is probably, I think, the hardest step, engineering the bacterial infection processes. And certainly in all these nitrogen-fixing symbioses that lead to nodules, you always have the bacteria colonizing the cells of the plant. So I think that that's a very, very important process to have the bacteria inside the cells of the plant. And getting the bacteria to colonize the plant, and particularly if you're going to use these infection structures, that's probably, for me, I think, the hardest challenge that we face. And ultimately, once you have the bacteria inside the cells of a dividing organ or an expanding organ within the root of the cereal crop, you have to, in that environment, provide the environment for nitrogen fixation. And most of that is about regulating the oxygen levels that allows the nitrogenase enzyme to function, and also exchanging the nutrients between the plant and the bacteria. So I wouldn't say that we're at a stage where we can engineer all of these processes into a cereal. And I think that the way I think about it is, we've made massive leaps in our understanding of how rhizobial recognition occurs in legumes in the last decade or so. And that's because of forward genetics in the model legumes have given us the signaling pathways that allow the recognition of the rhizobia. These 2 processes, nodule formation we understand to some degree, but I wouldn't say we understand it sufficiently that I can tell you how to engineer a nodule. Bacterial infection is really lagging quite significantly behind. And that's not necessarily because you can't dissect bacterial infection by genetics. It's just that I don't think the community has focused specifically strongly on the genes that are required for bacterial infection. So this process is somewhat lagging behind, and I really cannot tell you how we would engineer bacterial infection at this stage. But that's not to say that we shouldn't start this process of engineering. Engineering nitrogen fixation into cereals is going to be a long-term project. I hope I can achieve something within my research career. Maybe I can't. Maybe I'm going to have to pass the baton on to somebody else. I hope that by the end of my research career, we have something that's usable, and particularly something that's usable to smallholder farmers. But I don't think you should not start this engineering process because you don't understand all that. I don't think you should go through a legume and say, okay, we know everything now, and now we're ready to engineer into cereals. My argument is that we can start this engineering process, and in parallel, the whole scientific community, or at least the legume scientific community, expands our knowledge in some of these other areas. Okay, so the first step, which is a step that I've been focusing on, so in the Gates-funded program, we are just focusing on this first step, engineering rhizobial recognition into cereals. And that is really trying to replicate the chemical communication that happens between the legume root and the free-living rhizobia in the rhizosphere. And in particular, teaching cereals to recognize this signaling molecule, nod factor, that's coming from the rhizobial bacteria. And about a decade of research of genetic dissection in legumes in the model legume Medicago has given us a signal transduction pathway that we know is responsible for perceiving nod factor. So we have receptor-like kinases sitting on the plasma membrane. They transmit the signal to a number of channels that are sitting on the nuclear membrane. They're involved in the activation of calcium oscillations in the nucleus. And then we have a calcium decoding complex that's driven by this calcium and calmodulin-dependent protein kinase that regulates gene expression, and they activate a suite of transcription factors that in turn activate additional transcription factors, and ultimately you have a network of transcription factors that are coordinating the gene expression changes, not only appropriate for activation of a nodule, but also bacterial infection. So with the knowledge that we have on the signaling pathway in Medicago, our question was, can we engineer this signaling pathway into cereals to allow nod factor recognition in a cereal? Now, what helps us a lot in that process is the fact that this is a bifunctional signaling pathway. It's not only, in the legumes, it's not only necessary for perception of nod factor and activation of nodulation, but it's also required for the perception of mic factor, which is a signaling molecule that comes from arbuscular mycorrhizal fungi. And arbuscular mycorrhizal fungi have many parallels to the nitrogen-fixing symbiosis. There's a lot of genetic parallels, but there's also many similarities that we see in these two processes. So in both cases, the bacteria or the fungus is free living in the soil, and in both cases, you have to activate this chemical communication that allows the establishment of the symbiotic associations. And also what's true in both cases is that ultimately, it's an intracellular accommodation of the symbiont. So the bacteria are contained within the symbiosome, and the fungus is ultimately forms these arbuscules, which are contained within the plant, inside the plant cell, and surrounding all of this fungal hyphae is plant membrane. And also, both symbiotic associations are associated with the exchange of nutrients. So if we look at the evolutionary history of these processes, nodulation emerged about 60 million years ago. It was right at the base of the legume family, but it really is restricted, and there's multiple independent origins of the evolution of nitrogen-fixing symbioses that lead to nodulation. So we've got the legumes, but we also have a number of species in the actinorhizal clade that form nitrogen-fixing nodules. And all of those are contained within the supergroup, the urosids. In contrast, mycorrhizal symbiosis goes right back to the very early stages of the evolution of higher plants. It originates to about 450 million years ago, and we know that because we have a fossil record that documents arbuscular mycorrhizal associations in the very earliest land plants. So as a result, arbuscular mycorrhizal associations are really ubiquitous within the plant kingdom, and if there's plant species that can't form arbuscular mycorrhizal associations, such as the brassica, it's because they've lost it, not because they never had it. So the brassicas, ancestral plant species of the Brassicaceae, certainly had the capability to form arbuscular mycorrhizal associations. Now, importantly here, the monocots, which are the crops that we want to target for engineering nitrogen fixation, should in theory have we know have the arbuscular mycorrhizal association. And we know that if we mutate components of this symbiosis signaling pathway that was identified in legumes, so these are mutants in the sym pathway in rice, they abolish the capability to associate with the arbuscular mycorrhizal fungus. So here's a wild-type rice plant, and you can see it gets heavily colonized. All this dark staining is the fungus inside the roots. These are the vesicles that form, and you can see some of the arbuscules forming in the cortical cells there. And in contrast, if you mutate any of the genes in rice in the symbiosis signaling pathway, the fungus forms the initial stages of infection at the root surface but can't get into the root. So it forms hypopody on the root surface, but you don't get any colonization of the root. And that is phenotypes that are very similar to what we see in the mutants in legumes. What's more, we can take the genes from cereals, from rice, and transcomplement the legume mutants for these symbiosis signaling genes. So in this case, this is a mutant in Medicago that's defective in the calcium decoding protein, CCAM-K. We've taken the gene from rice, transformed it into Medicago, and you can see that this can now make very nice nodules, whereas normally this mutant is unable to make any nodules or even perceive the bacteria. So essentially what that is telling us is that the symbiosis signaling pathway is present in cereals. But what's more, it's also telling us that because we can transcomplement all of the components of the symbiosis signaling pathway from cereals into legumes and complement for nodulation, as well as arbuscular mycorrhizal associations, it's telling us that during the evolution of the process of nodulation, you haven't changed the core functionality of this symbiosis signaling pathway. So the pathway is there in cereals, and it's functioning in an identical manner to how it functions in a legume in its association with nitrogen-fixing bacteria. In other words, the activation of this pathway by either arbuscular mycorrhizal fungi or rhizobial bacteria must be equivalent. And from an engineering perspective, this is great news for me, because it means that I don't need to transfer the entirety of this signaling pathway. And if you imagine trying to engineer something as complex as oscillations in your nuclear calcium levels, that is a pretty challenging thing to engineer. What we need to therefore focus on are the components that we believe are legume-specific. And certainly this group of receptor-like kinases, which are the lyse-M receptor-like kinases, they're massively expanded in the legume family, and almost certainly that seems to be associated with expanding the number of polysaccharide signaling molecules that legumes can perceive that allowed the perception of rhizobial bacteria. And we also see we believe that a number of these transcription factors that sit downstream or in parallel to the symbiosis signaling pathway are also nodulation-specific or have nodulation-specific functions. Okay, so from an engineering perspective, we are not focusing on this whole pathway. We're just focusing on the immediate components upstream of the pathway and the transcription factors downstream of the pathway to try to transfer this capability to perceive rhizobial bacteria. So as a starting place to this engineering process, I felt anyway that what we really needed to understand was how this signaling pathway was functioning in a cereal. And if we wanted to understand that, we had to understand how the signaling pathway was functioning as the plant forms the association with arbuscular mycorrhizal fungi. So this is although the signaling pathway was discovered because of its interactions with rhizobia, really its native state, or the place in which it evolved and the function that it plays in most cases in the plant kingdom, is right here when the arbuscular mycorrhizal fungus is making contact with the root of a plant, and in particular forming these highly branched hyphae that are associated with the early colonization of the plant root. So these highly branched fungi will grow down towards the plant root and then will make hyphapodia on the surface of the plant root and then colonize inside the plant root through the epidermal cells. So it's at this stage where this pathway, which is when we need to understand what this pathway is doing in the context of arbuscular mycorrhizal fungi. And although for many years we've known the structure and the nature of nod factor, we haven't known what the signaling molecules were that were coming from mycorrhizal fungi. A few years ago, it was discovered that mycorrhizal fungi did actually make lipoketooligosaccharide signals, and these look very, very similar to the signaling molecules produced by rhizobial bacteria. So these lipoketooligosaccarides have a chitin backbone, an acyl chain, and then in some cases additional modifications. But in the mycorrhizal fungi, we saw very, very simple lipoketooligosaccarides, so not sulfated, not acetylated, just basically a chitin backbone with the acyl tail. It's since the publication of this paper been shown that there's actually quite a variety of different lipoketooligosaccharide signals produced by arbuscular mycorrhizal fungi. Now, in addition to this, it was also demonstrated that mycorrhizal fungi make very simple short-chain chitin oligosaccharides. That's perhaps not very surprising because chitin is the essential component of the cell wall of fungi. But it was proposed that both LCOs and COs could act as the so-called MIC factors that act as the signaling molecules coming from arbuscular mycorrhizal fungi. So we use the oscillations in calcium as a way to measure the capability of cereals to recognize a whole slew of different signaling molecules, of these LCO and CO signaling molecules. And we tested, these are the mycorrhizal produced LCOs. There's two that we're testing here coming from arbuscular mycorrhizal fungi. And we also used some species of rhizobial bacteria that make a very large spectrum of lipoketooligosaccarides. So we could test many, many different lipoketooligosaccarides in these preps from these broad host range rhizobial bacteria. And you can see that for the LCOs, we were not able to see any responses in rice. We've since repeated this also in barley, and we've yet to see any activation of this signaling pathway by the LCOs. But in contrast, the COs, CO4 in particular, activates this pathway very well in rice, and we've also seen it now in barley. And if you just summarize this here on this table, you can see this is looking at the percentage of cells that are activating for calcium oscillations, which we're using as our marker for activation of the symbiosis signaling pathway. The CO molecules activate calcium spiking, but or none of the LCO molecules are able to do so. And now if you compare a legume to a non-legume, so here we're looking at Medicago and lotus, the two model legumes, in contrast to two non-legumes, tomato and rice. You can see that legumes have the capability to perceive the lipoketooligosaccharides as well as the chitooligosaccharides, but non-legumes pretty well, with a few exceptions of very low responses, generally are perceiving the chitooligosaccharides and not the lipoketooligosaccharides. And here we're using very high concentrations, so this is 10 to the negative 5 molar, extraordinarily high concentrations of these signaling molecules. We can go down, with the COs, we can go down comfortably to 10 to the negative 9 molar and still see the activation of calcium oscillations. So we can certainly go down to much more biologically relevant concentrations, but in this analysis we wanted to use very high concentrations such that if we did not see a response to the LCOs, we were confident there wasn't a response there. Okay, so that highlights, if I just go back to this, it highlights that probably one of the key differences, or one of the key innovations that happened in the evolution of nodulation in legumes, was the capability to expand the profile of polysaccharide signals that could be perceived and that could activate the symbiosis signaling pathway. And for me, it highlights that if you're going to transfer the capability to interact with rhizobial bacteria, you have to get cereals to start recognizing these lipoketoooligosaccharides, which are the predominant signaling molecules produced by the nitrogen-fixing bacteria. And therefore, in the Engineering the Nitrogen Symbiosis program, we have really a number of objectives, particularly for the engineering objectives here I'm just showing you. So the first is engineering the nod factor perception. And for this, we don't have to rely on the activation of some process downstream in nodulation. We can just look for the activation of this pathway using calcium oscillations. So can we now, when we transfer these receptors from the nod factor receptors into cereals, not only see the activation of the pathway by chitooligosaccharides, but can we now activate the pathway in response to lipoketoooligosaccharides? And in addition, can we engineer this downstream components of the pathway, and in particular, the suites of transcription factors? Can we get these suites of transcription factors to function in cereals to activate their cognate reporters? Okay, so if we just demonstrate, this is really what we're attempting to engineer. The receptor complex, this is the receptor complex that we know about that's associated with perception of nod factor. We have two nod factor receptors, NFR1 and NFR5, that have both been shown able to bind nod factor directly. And they function in conjunction with a co-receptor, SymRK, that appears to be involved in producing secondary messengers to activate nuclear calcium oscillations. Now, we don't yet really know what the MIC factor receptors are, but we do know that SymRK is part of the common symbiosis signaling pathway. So SymRK is the first step in the conservation between perception with rhizobia, interactions with rhizobia and interactions with arbuscular mycorrhizal fungi, suggesting that SymRK must form a receptor complex with probably lysem receptor-like kinases that are involved in perceiving chitooligosaccharides or the MIC LCOs, and then activating the secondary messengers to give downstream nuclear calcium oscillations. So this suggests that really what we need to be able to do is transfer these receptors, have them expressed in the right cells at the right concentrations present on the plasma membrane, and we hope that they will already form complexes with the SymRK, that native SymRK present in cereals. And then downstream of calcium oscillations, as I said, we've got this calcium and calmodulin independent protein kinase, interacts with and phosphorylates a transcription factor cyclops. That seems to be one of the first steps in the activation of nodulation-associated gene expression. But in parallel with and downstream of, we have suites of transcription factors that are coordinating gene expression downstream of the symbiosis signaling pathway. And all of these are identified for mutant analysis, and we know they're all key for activating the different processes associated with nodule organogenesis and bacterial infection. And as we get further and further down this pathway, we can start to break apart the genes that are necessary for bacterial infection versus nodule organogenesis. Okay, so one of the first challenges that we had was what plant to engineer. So the Gates Foundation insisted that we work on maize because it is the #1 cereal crop for smallholder farmers in Africa. So our target here is maize. The BBSRC funding supports work also in wheat, and anything that we do in maize we'll also do in wheat. So we have maize and wheat running in parallel. But neither maize nor wheat are very good tools for engineering. You would hate to have that as your primary target. Maize is extraordinary. Most maize and wheat are extraordinarily expensive to transform and extraordinarily expensive to run GM maize and wheat in the field. And you'd have to do it all in the field because bulking seed in the greenhouses with these large plants is just impractical. So we started off by saying, okay, we're going to use one of these model cereals, and we focused on Soteria. And the reason we focused on Soteria was because it's a C4 grass. It's developing as a model cereal for the larger group of grasses that includes maize, and it seemed like it was certainly developing as a reasonably good model. But we spent 2 years struggling with Soteria, and the challenge for us was the transformation efficiencies. We were getting when we started off, we got getting transformation efficiencies of about 0.5%, which was just not workable. We managed to get that up to about 5%, but that still wasn't workable. So that's 5% of the calli that you transform will give you transgenic plants. That was just not a workable. We've got to put in hundreds of constructs into these plants, and we just couldn't work with such low transformation efficiencies. In contrast, transformation efficiencies in barley are somewhere in the range of 25 to 50%. On a good day, it's 50%. On a bad day, it's more like 25 to 30%. And so we've actually made the transition. After 2 years of battling with Soteria, we gave up with Soteria and focused on barley because we had a very good barley transformation pipeline here at John Innes Center. We could work with barley relatively easily here at the JIC. The other obvious one that we could have done was rice, but a lot of the research that's happening is in more temperate countries. A lot of the research happening in this program is in more temperate countries, and actually handling thousands and thousands of rice transgenic lines is somewhat impractical when you have to, in the UK anyway. So therefore, at the moment, our model cereal is barley. I wouldn't have started this project expecting that we would have had barley as our model cereal, but it's actually working extremely well for us. Okay, so we've got barley as our model cereal, and the other thing that we put into place was Golden Gate cloning. And Golden Gate cloning had been around for a few years, but nobody had really been using it at that sort of very high throughput and at a capability that we could bring the costs down for Golden Gate cloning that allowed you to make many thousands of constructs. So without going into the details of what Golden Gate cloning is, it basically treats everything as modules. So you treat your promoter as a module, you treat your open reading frame as a module, your terminators as modules, and it allows you to mix and match any of those different modules. And then once you've made a synthetic gene, so say with a specific promoter, a specific open reading frame, a specific terminator, that gene itself can become a module that you can then build together these large transformation vectors, constructs. So the great thing here was it gave us total flexibility. We used Gibson assembly to create our modules, and once we had those modules, we could mix and match any gene with any promoter, with any terminator, and then mix and match any genes with different reporters in the constructs. And when we put this in place, when I wrote the grant originally, and I look back at it now, I think, wow, that was incredibly naive, because we said we'd make somewhere 20 to 40 constructs and transform them into cereals in the original proposal. Once we got Golden Gate in place, we could make 100 constructs in a week. Just one postdoc could make 100 constructs in a week. It totally trivialized construct production for us. Okay, so here's an example, and I just want to give you some example. I'm just going to focus on the engineering of the pathway sitting downstream of calcium oscillations. We have put in about 250 constructs into barley now, which equates to about 2,500 barley transgenic lines that we have to work with. And it is a work in progress. This is just a progress report at this stage. And at the moment, we're wading through thousands of T1 lines to analyze the effects on the symbiosis signaling pathway. And I'm just going to focus in my talk today just on the components sitting downstream of calcium oscillations. So we have this calcium module independent protein kinase that activates a transcription factor. But actually, this module is already present in cereals because it's part of the the common symbiosis signaling pathway. The modules that are not present in cereals, or we don't believe are present and functional, are the NSP1, NSP2 that drives the expression of this gene ERN. ERN itself is a transcription factor that then binds to what we call the NOD factor box and activates a suite of genes associated with NOD factor perception. And then NIN, which is a gene that's activated by this calcium decoding complex, is a key transcription factor that goes on to coordinate quite a lot of the genes associated with nodule organogenesis. Okay, so we can break this down really into component parts. And the first experiment I'm just going to tell you about is NSP1, NSP2, driving the expression of ERN, and then ERN itself driving the expression of another NOD factor reporter, the NOD factor box. So this is a sort of things that we were doing. So when we do an experiment, we basically make a number of constructs, maybe in the beginning, maybe 5 to 10 different constructs. And those constructs created one experiment. So all of those constructs going to barley, we have all the controls intrinsic to that experiment. And then we just ask, can we engineer this module into barley? And in the first instance, we were very conservative. So we used very well-known, very well-established promoters that were ubiquitously expressed. So here's the maize ubiquitin promoter. So very high expression. We know it's in many, many different tissues. And although we ultimately do want root expression, in the first instance, we just wanted to ask, can we transfer modules from legumes into cereals and get them to function? So in this is essentially, we have a number of different constructs here, and you'll see the different controls. But the end result that we're trying to achieve is express NSP1 and NSP2 that function as a dimer, the tetramer actually, to then activate this promoter, the ERN promoter. ERN itself is a transcription factor that then can bind to the NOD factor box and drive the expression of GUS. So in this, we wanted to really not just transfer one module, but really start to build a slight network of transcription factors to coordinate processes in the cereal. So firstly, these are all the different constructs. So in this experiment, we had 6 different constructs gone into barley. Each of these is an individual barley line. So we're looking at T1 seed. So it's segregating, but we just focus on the homozygous plants in the segregating. So each of these is an individual barley line that has been transformed. And first of all, we're just looking at the genes that we've engineered in. So here's NSP1 expression, and you can see that when we engineer NSP1, pretty well all the lines that have NSP1 are expressing NSP1. Equally, all the lines that are engineered with NSP2 are expressing NSP2. So the promoters are working, the Golden Gate cloning is working, which was a relief. And then, so the first question is, can we get the expression of the reporter? And in this case, the first reporter is the ERN reporter, so that's the ERN1 promoter driving the expression of ERN. And very happy to say that actually NSP1 and NSP2, when transformed into barley, seem to be driving the expression of ERN in some of the barley transgenic lines. And we see reasonable correlation between the expression of NSP1 and NSP2 in these lines and the lines that give us good ERN expression. So then the question is, can we then get the next step? So can we have a little mini network working in cereals? And it's not as convincing, I've got to say, and we've really got to go in and validate this, but it does look like ERN is driving the expression of the NOD factor box as well. So we are seeing some, at least higher levels of GUS in the lines that are expressing where we know ERN is expressed. So it certainly seems that we can start to build legume-specific networks in cereals and have them activating legume-specific NOD factor promoters in the cereal crops. So it certainly seems that we've definitely got this module to work and drive the expression of a legume-specific transcription factor in cereals. And then it seems, although the efficiency is not as good, but we seem to have also engineered the next step, which is the ERN activation of this legume-specific reporter for NOD factor activation. So that's just looking at this module here and this module here. Now, actually, one of the main targets that we were attempting to engineer was CCAM-K, and because we have gain of functions in CCAM-K that bypass all the need for the activation of the early parts of the signaling pathway. So we wanted to get that gain of function in CCAM-K to activate at least a reporter, the NIN reporter, and then look at the suite of genes that get activated in cereals when we get that CCAM-K gain of function to work. But we've done quite a bit of engineering, a lot of different lines engineered with CCAM-K gain of functions, and we've not been able to see any successful engineering, even though we're using some of the same promoters that I've just shown you to demonstrating activation of NSP1 and NSP2. So in some cases, the engineering works, and we get the modules working as we expect they should be. In other cases, the engineering isn't working, and we don't know. Why is that? Are we using the right promoter? What is a good promoter for symbiotically relevant expression of these genes coming from legumes in a cereal? And actually, one of the biggest challenges for us in this whole process is knowing which promoters we should be using. So we've done an awful lot of this, promoter GUS analysis. This is promoter GUS analysis in Medicago using a suite of different root-specific promoters. We've recapitulated all of this now in barley. I can tell you exactly which of the promoters that we've tested give GUS expression in barley. But I don't know, is that a good level of expression for a symbiotic receptor? Is that a good level of expression? Or maybe it's actually something very, very subtle. It's very difficult. Just because you see GUS expression doesn't necessarily tell you that that's the promoter that is going to work for your engineering. So we've also done this in our analysis of promoters. So here we're taking a whole suite of different promoters, primarily from cereals, and asking if they can complement the CCAM-K mutant when they express CCAM-K in a legume. So we're looking at complementation assayed by number of nodules that form on the plants, and all of these constructs are identical except for the promoter that drives CCAM-K. And you can see some promoters work very well. Here's the control is right here. This is the Medicago promoter driving CCAM-K, the native promoter. And you can see some of our cereal promoters work just as well as the native promoter of CCAM-K. Some don't work at all. So that at least tells us in a legume context that if we express these transcription factors from these cereal-specific promoters, we get symbiotically relevant expression. So, but it still doesn't tell us which promoters we should use for engineering. And so what we're now doing, because of the Golden Gate cloning and the flexibility that provides, rather than just using a very conservative promoter like the ubiquitin promoter, we've got 25 different promoters that we know can complement the genes in a legume, the symbiotic genes in a legume, and are expressed in cereals, and in some cases give us good levels of GUS, which is indicated by the blue staining in barley. But it focuses down from about 40 promoters down to about 29 promoters, but we don't focus down any more than that. We just make a whole load of constructs where we just mix all of the different, we use all of our promoters to drive all of the engineering genes that we want to. So rather than rather than saying, okay, we just use the ubiquitin promoter, we use all promoters to drive the engineering constructs. Now the challenge there is, you could do the same with all the terminators, and we've actually tested all of our different terminators, and some terminators work very well, some terminators don't work as well for the regulating gene expression. But you now have a whole suite of different promoters, a whole suite of different terminators, a number of different genes we want to engineer. The combinations that you can make become massive. And even though Golden Gate cloning is very easy, it actually still is pretty challenging to make all of these different constructs. So if you just look at how Golden Gate works, it's a modular system. You take all your different modules, you put them together into a level 1 module, and that level 1 module ultimately will come together into your binary vector. So when we do this by hand, there's a number of different processes that we use. We have to retrieve the modules from the freezer. That's actually very slow process. That's probably one of the slowest processes in this assembly platform, and also prone to human error. And then somebody sits there and pipettes them all. We do the digestion and the ligation. We do the transformation and the validation. But we can actually break these parts into individual components and basically replace the human with automation. Automated, so we now have an automated freezer that does that picks the modules. We have a echo liquid handler that can do all of the, putting all the different modules together into the plates, and then a robot that essentially, a Hamilton robot that does all the transformations for us. And when you do this, then actually there's no human involved. You can make constructs, you get your modules produced by a company. Nobody is making constructs anymore in my lab, or we've just put this into place. And this completely trivializes construct production. So we made 100 constructs in 8 minutes in the first round of this work. And once we have the platform running very smoothly, we can make thousands and thousands of constructs in a day. So certainly for this challenging engineering processes, this is the sort of scale that you need to be working at. But the challenge is the transformation. So you can make loads of constructs, but then you get to this massive bottleneck with the transformation. Ultimately, you have to get those into a plant in order to test. We could have taken a protoplast assay. I'm not very convinced that I'm going to engineer nitrogen-fixing cereals with protoplast assays. I really wanted to be looking in whole plants. And so the thing that we've done to really streamline the plant transformation is put in place what we call the rapid testing platform. And what the rapid testing platform is, still barley transformation, still stable transformation. But instead of thinking about you're taking all of your constructs through to T1 or T2, getting the constructs homozygous, you just make transgenic plants and you do destructive phenotyping on your transgenic plants. Now, if you do that, then you can have stable transgenic plants in 3 months. And you can also do it at a pretty high throughput, because the biggest bottleneck in transformation for us is not generating the transgenic plants, it's actually growing them all up to seed and then handling you get one construct, 10 lines, those 10 lines are all segregating and taking all of the genetics through to homozygosity when you've got hundreds of constructs and now thousands of constructs to put into the system is hugely challenging. So we take this, so essentially we generate 6 plants per construct. They're T0 plants. We say we don't want to take them to seed. We do destructive phenotyping on those T0 plants. And essentially what that allows us to do is first test the constructs, see if anything looks promising, and then any constructs that look promising, those are the ones we take through to seed and get into the T1 lines, and then we've got material that we can do much more involved phenotyping of. So one of the big challenges for us, we started off envisioning in the project making 20 constructs, putting them into plants and hoping for the best. And I realized very quickly that that's just not going to work, and it's certainly not going to work on an engineering challenge as large as what we tackle. So one of the big things that we've been doing through this program is just trying to put in place the scale of transformation and the scale of engineering that we need in cereals to really get the system up and running in cereals. So I think, I don't even know what my time frame was, but I think I'm out of time. So the question is, how close are we to engineering? How close are we to having nitrogen-fixing maize? Well, I would say we've got a lot of the platforms in place. That engineering technology, and particularly now with the tools that we have with synthetic biology, really putting us in a position that we can tackle some of these very, very challenging engineering problems. It's very early days for my project. But I hope that maybe in a year's time, once we've gone through this first round of barley transformation, I'm hoping that we can have more of the first step of the signaling pathway engineered into cereals. So I would say I'm optimistic that in the next few years we'll have pretty well the signaling pathway engineered into cereals. And I certainly hope that I put everything in place that's necessary for that. For the next 2 stages, nodule organogenesis and bacterial infection, we actually need to do more work, but that can run in parallel with the engineering processes, and ultimately, obviously, getting the right environment for nitrogen fixation. I acknowledge that's very important, but until I've got bacterial infection, I'm not taking that challenge. So, but just coming back to this graph, And as I said, if I'm going to replace fertilizers in totality, I have to be somewhere out here at the total level that a legume fixes nitrogen. But what I'm optimistic about is as we're going through these engineering processes, we can start to get useful levels of fixed nitrogen into cereals. And that's, when you say a useful level of fixed nitrogen, I'm imagining some rather imperfect looking nodule-like structure. It doesn't have to be as beautiful as a legume nodule to fix some level of nitrogen and to have pretty inefficient nitrogen fixation. But if I can get that, then I'm starting to impact quite dramatically on smallholder farmers and hopefully also gradually replacing the levels of nitrogen fertilizer that are used here in the developing world. So I hope I can get over here during my career, but if I get somewhere along here, then I'll be very happy. And I think we have a huge potential to have impact, particularly in the developing world, if we can get somewhere in this range of fixed nitrogen. So this is a reasonably sized international program that I run. I have collaborators with Jean Denare and Claire Goss groups in Toulouse. Jens Staugart is focusing on the engineering of the receptors, the nod factor receptors into cereals, and I didn't talk about that work, but we have some promising looking leads but that need to be validated in that area. Tom is managing all our maize transformation. I didn't talk about the maize transformation, but we have quite a lot of maize transformation happening alongside. Wendy is at the John Innes managing the barley transformation. And I should really acknowledge the Gates Foundation, in particular my science officer, Kathy Kahn. And I think without the Gates Foundation willing to take the risk, because of course engineering nitrogen fixation is very high risk, and it's difficult to persuade funding agencies to take that leap, but the Gates Foundation has been willing to take that leap, and I very much acknowledge that, the risk that they've taken. And within my own lab, Christian Rogers is a key player in developing the Golden Gate platforms and a lot of the synthetic biology tools that we're developing. The early work on the engineering was the work of Ben Miller. Now Doreen is doing most of the engineering work. And a support team or the calcium analysis done by John Ho's son. Andre, Eleni, and Andy as a support team for a lot of the engineering that's happening across the program internationally. And I'd be happy to take any questions.